gave me, allowing me to set up a lab doing structural neurobiology in Italy. So our main research goal is to try to understand molecular mechanisms underlying important processes in life. And the main research topic that we're trying to address is how do cells communicate. To do this, we basically investigate several pathways, and the one that I'm going to talk about today will be related to the pathway that leads to formation of neuromuscular junctions. In particular, our main focus is related to trying to answer to the three questions that you see on the screen at the moment, which is, what is molecular recognition? So how do molecules interact specifically? What is molecular regulation? So how do molecules in a pathway interact with each other, mediating their function and leading to the final events? And what are the elementary steps of a regulatory pathway? And this is a very important question, because if we know what happens in the homeostasis of a process, then we can design specific molecules to inhibit or activate pathways that are not working anymore. So, structural biology, and it is, I should say again that it is a great pleasure to be here, because I saw that during your symposium you had Ada Ionat, who was actually sending a message also before my presentation here, and she's also a structural biologist, so it is nice to share the same point of view in front of this audience. Structural biology is a technique that provides visual ways to understand what molecules do. We are crystallographers, so our main objective is to characterize how the atoms that form proteins, that form receptors, are actually engaged in their interactions with ligands. And I like to show you this image because I think that is a nice representation of the visual understanding of a very complex mechanism. This is a sculpture from an artist called Jan Tinguely. He's a Swiss artist who actually makes very complex structures and provides them with very interesting names. The name of this structure is the complex machine of life. And this is a nice way to represent what proteins, what receptors are. They are very complex objects that somehow are almost impossible to understand until we realize what are their constituents, how they are made, and how they precisely interact with each other. As I said, the main topic that we're trying to address is how nerves interact with muscles. And I don't think that I have to explain in front of this audience what neuromuscular junctions are, because I think that in a symposium where we discuss spinal cord neuroscience, I think that this is just too much, so I will go quickly on this. But basically, just to make a long story short, what we're trying to understand is how the motoneural axons are precisely innervating at specific points on the surface of the muscle cell membrane. Okay, so this is a picture obtained from microscopy related to neuromuscular junctions. I think that many of you have seen pictures like this, and I just would like to emphasize the specific connection points that we're seeing. So the striped background that you see is basically related to the muscle fibers, and this sort of tree-like structure that you see is coming from the motoneural axon that's been stained, and the leaves that you see from the tree are the neuromuscular junctions. So if we look at them using fluorescence microscopy, they can be observed with this pretzel-like structure. They have been defined as pretzel-like because of this complex organization that they have. This is a staining obtained with bungarotoxin, actually, that shows the complexity of this architecture at the interface between various cells. So at the molecular level, what do we have? Basically, we have this complex corrugated surface that is surrounding the point of connection of the motoneural axon. And at these positions where the connection is present, we have a very intense clustering of receptors. And as we are molecular scientists, I like to show you some electron microscopy and some three-dimensional structures of the acetylcholine receptors, which is the main receptor that is located at the contact point between neuron and muscle cells at neuromuscular junctions. So here you can see a better three-dimensional representation in cartoon of the acetylcholine receptor, and you can see how large the molecule is. This is a pentameric ion channel composed of a large extracellular region that is responsible for capturing the signal from the interface between the cells, and then through a complex process of crosstalk, it delivers the chemical message into the muscle cells, basically by providing a gradient of ions. So, in a very simplistic view, this cartoon shows how the acetylcholine receptor works. The acetylcholine receptor, the main neurotransmitter, binds to the surface, and the opening of the ion channel basically results in channeling of ions into the cells. The overall muscle fiber depolarizes, inducing contractions and allowing the movement. So a very intriguing question that we're trying to address is what happens when the innervation occurs? Because the innervation of the axon at precise position depends on the clustering of receptors. So the formation of, the formation of these compact structures on the surface. But at the same time, we have a sort of chicken and the egg problem, because the clustering of the receptors depends on the presence of the motoneural axon, in particular of ligands coming from the basal lamina surrounding the axon when the interaction occurs. So, how an event that is triggered by the presence of the axon allows the binding of the axon on the same site? Well, what we're trying to investigate is basically how this series of positive and negative signals triggering the clustering of receptors on the muscle cells occurs. And in particular, we're focusing on the positive signal. In short, the Aegerin signaling. So, if we look in detail at what we have on the surface of the muscle cells, we have what we like to call a forest of receptors, because we have a very complex distribution with many molecules, ligands, receptors, effector molecules, regulators, inhibitors of the interactions. Obviously, as our focus is molecular, we cannot study the whole system throughout. We would need probably tens of PhD students and postdocs in the lab, and this is something we cannot afford. So we restrict our focus to the most well-studied system and to those that we think are the most interesting ones. And in particular, we focus on the Aegerin-LRP4 complex, that is the one highlighted here on the left, and the mechanisms of collagen signaling, which are becoming a more and more hot topic in neuromuscular junction signaling. I would like to focus a bit on the Aegerin signaling processes. So, if we look in detail at those 3-4 molecules that are essential for the process, we basically see that we have a receptor tyrosine kinase that is located on the surface of the muscle cell that interacts with a large co-receptor molecule that has been recently identified, which is called LRP4. Upon signaling from a proteoglycan coming from the basal lamina called Aegerin, LRP4 changes its conformation, and in a process that is yet unknown, and this is one of the main research questions that we're trying to address, changes its conformation, activates the MuSK receptor, and induces an initial autophosphorylation. This autophosphorylation, through a pathway that involves many different signaling molecules, induces activation of a receptor-associated molecule called rhabsin, and rhabsin itself facilitates the aggregation of acetylcholine receptors on the surface of the muscle cells, enabling the axonal innervation that we saw before. Another important player that we're trying to investigate at the moment is called neurotrypsin, which is a trypsin protease-like specific of neuromuscular junctions. So this molecule here is known to process Aegerin by cleaving it into 2 fragments, and the C-terminal fragment of Aegerin that is released is still capable and competent for MuSK signaling, but somehow it seems to be implicated as well in a series of diseases, including important syndromes associated with aging, such as sarcopenia. So, in fact, the fascination that we have for this molecule goes far beyond the basic research, so the understanding of how this molecule looks like, but it is addressed on understanding why specific genetic mutations and specific autoimmune diseases associated to these molecules actually induce highly invalidating diseases in the human body. And in particular here you can see a list of diseases and cellular events that are associated with the molecules that are listed in this picture. So, the main questions that we're doing, that we are addressing in the lab, and the main focus is how do LRP4 and AGRI interact? How is MuSK activated? So how is the receptor through the interaction of the ligand with the co-receptor activated, inducing the process of clustering of the acetylcholine receptor? And then how do these molecules that are located in the extracellular space are interacting and then sending the signals through the intracellular part? And then what are the functions, the specific functions of the rhabsine molecule with respect to the clustering of the acetylcholine receptor? As I said, I do not have many results, but I would like to share with you some unpublished data, which I summarized on this slide. A standard structural biology project is something that requires many years in order to obtain results that can provide an understanding at a more applied research level. But what I can share with you is that we, at the moment, managed to produce all the targets that we are investigating, and we are producing them in large amounts. For example, you can see here the large amount of recombinant MuSK receptor that we managed to produce in a well-folded way. You can see here an image of rhabsine localization on the surface of HAC293 cells that have been co-transfected in the presence of acetylcholine receptors, showing that we basically managed to reproduce a system where we can study the precise effects of rhabsine and of our system inside a model cell, a model in vitro system. We are doing electron microscopy on the MuSK LRP4 complex. And the image here, unfortunately, has not the resolution that you would expect to understand something, but if you trust me, you can see some worm-like structures that represent the interaction of the extracellular domains of the two receptor-co-receptor system together. We are doing binding studies to quantify how the molecules are interacting with each other in vitro. So, and with this we are basically generating mutants associated to the genetic diseases, trying to understand what happens with respect to the interactions with the two molecules together. And obviously, as we are structural biology and this is our core business, we are approaching the structural characterization of these molecules. And here you can see two fragments of a proteoglycan that is very similar to AGRI, that is specific to Ripon synapses, that is currently subject of investigation in the lab. So I would like to close my presentation by thanking the people that next to me are doing most of the work in the lab, so my postdocs and my PhD students. You see that I put them close, each one to one of the precise components that we're studying, because basically the lab is tackling the whole pathway in a sort of compartmented way. And we hope that our results taken together will give us a better understanding of the whole system at the molecular and possibly at the cellular level. And with this, I would like to thank you very much for your attention, and I would like to thank all the funding bodies that are providing our group with the resources to conduct our research, so the Harvard Foundation, Fondazione Cariplo, and remembering Rita Levi-Montalcini, also the Rita Levi-Montalcini Fellowship that I got last year. Thank you very much.